All right. So moving on to our next segment. We all know genes only pull the trigger. What kind of environment you're exposing your body to decides whether those genes will be manifested or not. It's all about the science of epigenetics, and in functional medicine, the entire focus is on epigenetics. So we have our next speaker, Dr. Shelly Mahajan. She's a leading pathologist and molecular specialist in cancer diagnostics. In her session, she will explore the pivotal roles of genetic mutations and epigenetic modifications and in the onset and progression of cancer. She'll discuss on how these insights can pave the path for innovative approaches in early detection, personalized medicine, and targeted cancer therapies. A warm welcome, Dr. Mahajan. Over to you. Screen.
Yeah. Then I'll have to tell you to go to the next slide every time.
Yes.
Let me just try once again because I just think it'll be a little uncomfortable. Just a second. Just let me just try once more to share my screen only.
You can also go to the slideshow menu and go for slideshow. There's a menu.
Okay. Yeah.
You can go from beginning.
Now is that okay?
Yes, it is.
Okay. I'm so sorry to take this time. Good afternoon, everyone. I'm Dr. Shelly Mahajan. I am the lab director at Mahajan Imaging and Labs. I'll be speaking today, and it's an honor to be speaking right after Dr. Thomas. It was a very enlightening presentation, and I'm sure nobody wanted him to stop. So I hope I can match up to his standards to some extent. I'll be speaking on genetics and epigenetics, and as we know today with all the cancer research, genetics and epigenetics are the two major mechanisms behind cancer development. And it's important to understand genetics and epigenetics to understand the pathogenesis of cancer, which in turn helps in improving and working on cancer treatment options, improved options. So I'll just to give a basic of what is genetics and what is epigenetics. If we go by the book definitions, WHO says that genetics is the study of hereditary. National Human Genome Research Institute says that it's a branch of biology concerned with the study of inheritance, including the interplay of genes, DNA variation, and their interactions with environmental factors. While NCI says that it's the study of genes and hereditary. Heredity being the passing of genetic information and traits, like as simple as eye color, from parents to offspring. So just to give one meaning to all these definitions, genetics essentially is the study of genes and mutations affecting the DNA sequence. Right, so DNA sequence, as we know, is the sort of the defining factor of everything, that is every trait that we have, be it normal or abnormal. So what is DNA? In DNA, what we are looking for is the order of just these four nucleotides: A's, T's, C's, and G's. And the sequence of these nucleotides, the sequence in which they are present in a particular DNA, it gives the information and it encodes for a protein, which in turn affects our physiology. So like in any word, if basically, if the sequence of any of these alphabets is changed, it changes the meaning of the word entirely. In DNA sequencing, what we do is we basically determine the exact order of these bases in a strand of DNA to identify whether the sequence is normal or abnormal. And the purpose of doing this is to search for any genetic variations or mutations that may play a role in the development or progression of a disease. So now what is epigenetics? Epigenetics, again, taking book definitions, the study of changes in gene function that are mitotically or meiotically heritable and that do not entail a change in DNA sequence. Also, epigenetics is defined as a field of study focused on changes in DNA that do not involve alterations to the underlying sequence. And it refers to the way your behaviors and environment can cause changes that affect the way your genes work. Now, simply put, epigenetics means the study of heritable changes in gene expression without DNA sequence alteration. Now, coming back to genetics, genetics means that there is a variation in the gene sequence. It may be heritable, it may be passed on or not passed on. Epigenetics is basically changes in gene expression without sequence alteration. Now, both these mechanisms have been considered to be, have been now found to be equally important in pathogenesis of any disease and for today's purpose of today's discussion, particularly cancer. In DNA sequencing or in any DNA or RNA sequencing, essentially the steps of the process are that we basically extract the nucleic acid, be it DNA or RNA, and then we prepare multiple copies of those DNA fragments, and then the libraries are prepared. Then it's put in a sequencer where the machine gives us the data, the bioinformatics data, wherein we know in that particular sample, it may be tumor sample, it may be a normal sample, it may be a blood sample, whatever that nucleic acid is. It gives us a sequence of the four bases the DNA bases that we spoke about. Now, our purpose is then to sort of match the DNA sequence of that particular sample with a reference genome and identify whether there is any abnormality in that sample or not. Now, like I said, these are just for example A, B, C, D, and E. There are no just to explain. If when we compare the sequence with the reference genome, we can identify what is the abnormality in that sample. Like if a base is deleted, it's called deletion. If an extra base is inserted, it's called insertion. Similarly, duplication, inversion, translocation. So we identify these changes which are called as mutations in the sample, and then we clinically identify them as benign and disease causing. Now, why? Of course, every mutation is, no two DNAs are the same, right? So brown eyes, black eyes, curly hair, straight hair. So we sort of identify whether a particular mutation, the difference from reference genome, is disease causing or not. So coming back to cancer, after these basics of genetics and epigenetics and DNAs, cancer, is it a genetic disease or not? I would say yes. Yes, cancer is a genetic disease. It is caused by changes in genes that control the way cells grow and multiply. Cells, we know, are building blocks of body. Each cell has a copy of your genes, which act like an instruction manual for the entire set of our expressions of the expression of every protein, every function of every cell. So yes, cancer is a genetic disease because it is caused by some mutations, some genetic mutations in our DNA or in the genes. Those mutations can be heritable or non-heritable, can be passed on to the offspring or not passed on. But the eventual pathogenesis, if we boil down to it, is some sort of genetic or epigenetic change happening. But is cancer a hereditary disease? It can be a yes or a no. Cancer can be passed on from one generation to another or cannot be passed on to one to the next generation. Now, how do we differentiate that? There are two kinds of mutations we are talking about, either germline or somatic mutations. A germline mutation is something which is there, which is present from the at the embryonal level, right? So because it is there in the embryonal level, it is present in all the cells of the body. And because it is present in all the cells of the body, it can very easily be identified by a simple blood sample, because from the white cells also we can take out the DNA, right? And also because it is embryonal, it can at present at the germ cell level, it is passed on from one generation to the next. So these are called as germline mutations. Germline mutations are basically help us to know the inherited risk of developing cancer for a specific individual, and hence is used in predictive medicine. versus somatic mutations. Now, somatic mutations are something which are acquired through our lifetime because of certain carcinogens. It can be environmental carcinogens, it can be chemical carcinogens, it can be smoking. You know, I mean, any carcinogen, any outside carcinogen, if a mutation is caused because of that, which leads to a cancer, it is called a somatic mutation. Now, because it is not present in germ cells, it is not present in all the cells of the body. It is only found in the tumor DNA. So, and that's why the most effective way of finding that mutation, of identifying that mutation, is testing the tumor DNA. It can be a direct histopathology of the tumor, or from a liquid biopsy done on the blood sample. But because it is not present in all the cells of the body, it is not present in the germ cells, it may not be necessarily transferred from one generation to the next. So somatic mutations are used to know which drug is most suited for this particular patient. So it's called precision medicine. Now, just to give an example of the genes which are affected in these mutations, germline and somatic, I'll talk about the most commonly talked about or most famous gene, which is the BRCA gene. It is the name comes from breast cancer gene, which is a misnomer because this gene may be mutated in other cancers as well, like ovarian, pancreatic, prostatic, melanoma. What we are trying to talk about here that it is a tumor suppressor DNA, tumor suppressor gene, and if mutated, its normal purpose is to suppress the tumor. If mutated, it causes cancer. It is unable to repair the damaged DNA, which is its usual function, and hence it causes cancer. Now, it is an autosomal dominant gene, so there is a 50, if one of the parents is affected, there is a 50% chance that it will go in the offspring as well. And then other kind of genes which may be affected for cancer formation is oncogenes. Oncogenes are normal purpose of an oncogene may be to stimulate cell proliferation when needed. But if it is activated, if it is a mutated oncogene, then it leads to inappropriate cell proliferation and hence cancer formation. Now, coming to epigenetics in cancer. So, epigenetics basically is changes in gene expression not caused by DNA sequence alteration, like we discussed. Epigenetic dysregulation is a common feature of many human diseases, particularly cancer. Now, defining the epigenetic defects associated with malignant tumors has become a focus of cancer research, resulting in gradual understanding of cancer cell epigenetic regulation. There are three mechanisms for epigenetics in cancer: DNA methylation, histone modification, and non-coding RNAs. Now, DNA methylation is, as the name suggests, adding a methyl group to the DNA. By addition of this methyl group, there is silencing of genes. Okay, now we'll go back to the other two mechanisms in a while. First, let's go deep into DNA methylation. The normal purpose of DNA methylation is to, it's basically vital for embryonic development, genomic imprinting, X chromosome inactivation, preserving chromosome stability. So this is the normal purpose of DNA methylation and gene silencing. But if it becomes aberrant, if it becomes abnormal, what will happen is there will be either excess methylation or less methylation. If it's hypermethylation, it can basically switch off, sort of silence or excessively switch off the tumor suppressor genes. And if that happens, we spoke about BRCA gene right now, there will be gene silencing and hence cancer. Now, if it's hypomethylation, that is, if the methylation is less than usual, then it can activate oncogenes. That is, it will stop the silencing of oncogenes when they are supposed to, and there will be genome-wide methylation. There will be methylation everywhere where it's not supposed to be also, and it will cause activation of oncogenes and immense genomic instability, which again leads to cancer. So this is the mechanism of aberrant DNA methylation causing cancer. DNA methylation in the promoter region of these are the examples for where aberrant DNA methylation causes cancer. For example, DNA methylation in the promoter region of retinoblastoma tumor suppressor gene, or repression of MLH1 expression by DNA methylation, which leads to genomic instability, which is a common mechanism like we know in gastric and colon cancers. Now, moving on to the next mechanism of epigenetics called histone modification. Now, histone modification is basically altering chromatin structure to regulate genes. The normal purpose of histone proteins is to keep keep the chromosomes in a compact, keeping it compact and to maintain their shape and protect the genetic material. Histone modifications control gene expression and other DNA related processes like repair damage or replication. So when this becomes aberrant, again, all this regulated process will become aberrant. The chromosome structure will become disheveled, and it will not remain in a compact state. So this deregulated gene expression, and which will lead to cancer cell proliferation, and cancer cell survival, metastasis, oncogenic transformation, all of this again it's understood to be now leading to cancer. Now, this is correlated with the etiology of a variety of human diseases, including allergic diseases, multiple sclerosis, GI cancers. Deregulation of gene expression plays pivotal roles in the formation of colorectal cancer. Like in this image, we can see there are these different majorly four process of histone modification, and essentially acetylation is something which deacetylation, loss of acetylation is something which is the most important factor in colorectal cancer formation. Thirdly, talking about non-coding RNAs, which are one of the RNAs which are formed after transcription. Non-coding RNAs are that part of RNA which is not involved in direct protein formation. Um, you know, as opposed to mRNA, for example. So they are not supposed to code for any protein function, but they do play a central role in regulating gene expression. Non-coding RNAs basically regulate gene expression post-transcriptionally. Now, aberrant non-coding RNAs, what do? They basically lead to they are dysregulated in cancers. The dysregulation of ncRNAs causes cancers, and this dysregulation can contribute to cancer development and progression. Non-coding RNAs can have both oncogenic and tumor suppressive roles, because they can basically either activate oncogenes or silence tumor suppressor genes, both. And because of this, they also have a major role in resistance to different cancer therapies. These are used as biomarkers to predict treatment response and detect resistance to therapy. So again, diagnostically, prognostically, for decision of treatment protocols, non-coding RNAs, which are again an epigenetic modification, becomes very important. Now, this is just wanted to talk about this because genetics and epigenetics cannot be, although are separate processes, but now we know, the studies have shown that genetics and epigenetics are not working in silos. There has to be an interaction between both these processes to lead to the pathogenesis of cancer and treatment of cancers. Genetics and epigenetics are intricately linked in the development of cancer. Like genetic modifications can alter the epigenome. Then epigenetic changes can sort of cause genetic mutations. These interactions play a critical role in cancer progression and carcinogenesis, contributing to uncontrolled proliferation or metastasis and cancer heterogeneity, right? Now, firstly, let's talk about how does epigenetics interact with genetics or affect genetic mutations. Epigenetic silencing, we know, causes loss of function of genes and predisposes to genetic mutations. Epigenetic silencing facilitates the selection of mutations. I'll talk a little bit about how this happens. Epigenetics can cause genes to be unregulated or silenced, which can lead to cell death or cancer. We know that. Again, epigenetic mechanisms can silence tumor suppressor microRNAs, which can be reversed with chromatin modifying drugs. And that's where the treatment part comes in. Um, uh, like for example, if we talk about MLH1, right? What happens in MLH1 gene mutation? It's basically hypermethylation of MLH1, which causes deficiency in the entire MMR mechanism, and hence causes colorectal cancer. Now, this process can be heritable or non-heritable, but it does cause has a role in pathogenesis of cancer. Similarly, if we talk about facilitation of and the selection of mutations. Now, sometimes what happens is MLH1, there are two alleles. One allele may be mutated already, and the other allele, because of epigenetic silencing, is silenced. So one process is facilitating the other process to and affecting the key signaling pathway of DNA repair and causing cancer, right? And then again, there are certain there are certain studies where the genetic mutations like P53, they are found in positions where epigenetic changes are common, right? So they occur at the sites of epigenetic changes and create a hotspot there. So, like I said, there is a very intricate and complicated interaction between genetics and epigenetics, and these two processes cannot be spoken about in silos. Now, again, microRNAs. Mutations in microRNAs can disrupt its recognition of binding targets and further result in oncogenic activation or tumor suppressor repression. Also, microRNAs target epigenetic modifiers, which lead to which affect or which increase future epigenetic changes. So, and then how does genetics affect epigenetics? There are these epigenetic modifiers which are involved in DNA methylation and chromatin remodeling and histone modification. Genetic mutations can affect all these genetic modifiers and in turn lead to an affected epigenetic machinery, be it DNA methylation machinery, histone modification, chromatin remodeling, and hence leads to deregulated gene expression and leading to cancer. For example, if we talk about the methylation genes, DNMT1, for example, if it is mutated, it will lead to, it can cause cancers like colorectal cancer. It's known to cause MDS or SNPs in breast carcinoma or lung cancers. So there is a very complicated mechanism and intricate mechanism where genetics can also affect epigenetics. Another example I'd like to talk about is IDH1 mutation. IDH1 mutation is very closely related to affecting this methylation process itself, the methylation modifiers, and it is known to be associated with glioma. Right. Now, talking about histone modifications, MLL1, 2, and 3. If there is any mutation in these modifiers, then again, it is known to be associated with bladder transitional cell carcinoma. So the idea is that these two processes, although have very distinct pathologies, but they do work in relation to each other in causing cancer. And hence, when there are researches happening on treatment part, there is not one mechanism which can be simply targeted to treat cancer. If we talk about genetic therapies, for example, if we know a specific mutation like EGFR in lung cancer, it's very commonly used EGFR inhibitors targeting specific mutations. Similarly, epigenetic therapies like DNA methylation inhibitors are now used commonly. Histone deacetylase inhibitors. Now, we also have some data on combined therapies, like that means integration of genetic and epigenetic approaches for better outcomes. Am I out of time? Okay, I'll just take 2-3 more. I'll just quickly run through the rest of the slide. So, just wanted to give an example of tumor suppressor gene being affected and leading to cancers like breast, ovarian, pancreatic, prostate. It is useful in identification of these mutations and predisposition to these cancers. And also, if it's a somatic BRCA mutation, we know that we can give a targeted therapy. Similarly, in ovarian cancer, if a BRCA gene is mutated, there is a targeted therapy available. We spoke about MLH1 mutation, which causes microsatellite instability and MMR deficiency. Again, it can be heritable, and both the processes, genetic and epigenetics, is involved in this instability. The identification of this instability becomes very important in identification and treatment also of Lynch syndrome cancers. Future directions, the identification of these processes is most commonly done by next generation sequencing. It can then multi-omics, involving multi-omics. That means integrating genomics, epigenomics, transcriptomics, everything together for diagnosis and planning of further treatment. And personalized medicine, where we can use genetic and epigenetic profiles together and tailor therapies according to that. In conclusion, cancer is driven by complex genetic and epigenetic mechanisms, and advances in understanding these processes are revolutionizing treatment. Future therapies aim to integrate genetic and epigenetic targets for personalized care. With this, I'll end my talk. Thank you so much, IAFM, for giving me this opportunity. Thank you.
Thank you so much, Dr. Mahajan. That was really informative. We'll take one question from the chat. So what's the incidence of MTHFR gene mutations in the Indian population?
MTHFR, it would be difficult for me to give the data right now, but MTHFR is it has been a rare mutation, but is again now coming up as we have more and more better identification methods and kits and reagents available, and knowledge of the mutation is more. It is on a rise in detection level.
Yeah. Okay. Yeah. Thank you. Thank you so much.